Good afternoon, ladies and gentlemen. My name is Dr. John Minatelli. I represent Valensa International. We're the human health company. Our company specializes in the development of clinically trialed, intellectual property protected, condition-specific dietary supplements. And today, it's my pleasure to introduce a new program that we have initiated at our company, which is to develop a brand new line of immune-based products. You might ask yourself, why would we want to do that? Well, according to a Nutrition Business Journal survey, the immune health category is one of the top 5 categories of interest to consumers. And this is validated by the fact that the total immune health supplement market was reported to be $2.3 billion in 2013 and was growing at a very healthy annual rate of 6 to 7%. And you can see this little figure here at the bottom of our slide. Basically, it's meant to show that our bodies are constantly assaulted by pathogenic bacteria, fungi, and viruses. And yet, on the whole, we remain healthy. But I certainly guarantee you that every one of you has been exposed and is also exposing others to pathogens all the time. And so this is a very important area, and it's an area which is still emerging in terms of science, the science of immunity. And just to show you what I really mean, here at Supply Side West, we have a total of 14,000 attendees, and they represent 60 countries. And during the course of the next few days, you're going to be meeting and shaking hands with everybody. If you take one of those hands and you put it into an agar plate, and then you simply let it sit overnight, that is what you get. So every one of us on our largest organ, which is our skin, carries millions and millions of potentially lethal pathogens. And our skin, by the way, has a very high level of immune cells in it. And this is what helps protect our largest organ from allowing us to fall into ill health or even dying from the presence of these particular pathogens. Interestingly, there are 4 main products that I would like to discuss very briefly today so you better understand what the U.S. market is all about. And they all arise from different circumstances in the business. For example, Airborne basically was a glorified vitamin for many years, and more recently has been fortified with beta-glucans, which are known immunostimulants. And during my talk, by the way, I may mention either the word immunostimulant or antigen. They mean the same thing. Emergen-C really was born by Linus Pauling, who was a very strong proponent of the use of vitamin C for the development of immunity. And more recently, both Wellmune and Epicor, which are yeast-based products, have been developed and are now the current standard of care in the business for true antigen-based products. But look what has happened in the marketplace over the last 3 or 4 years. Airborne was sold to Shift Nutrition for $150 million in 2012. Emergen-C maker Alicer sold its brand to Pfizer for $350 million in the same year. And even Wellmune, which is probably the most well-recognized product in the world, its maker Biothera sold its brand to the Kerry Group in 2015 for an undisclosed amount of money. And we owe a great deal of that to the people that have been involved in the development of these products. And these are really first and second generation products in the immune category. But they also suffer some pretty serious problems. And these are really the drivers that bring Valensa and its research and development team to take a look at what we can do to develop third-generation products. We have extensive commoditization now of the yeast-based beta-glucans in the marketplace, and that makes them ripe targets for displacement by advanced immunostimulants supported by well-designed clinical trials. And in this particular class, because we can't, as supplement providers, prevent or treat diseases, there are very limited claims that are allowable here, at least here in the United States, if not over the world. And if you take a look at the clinical work that has been done to support these existing products, it's very clear to all of us that most of the data is equivocal with respect to the claims that are actually on the package. And furthermore, some of the activities in terms of the primary endpoints of these clinical trials actually overstepped allowable supplement claims quite a bit. And furthermore, most of these clinical trials took almost a year to complete and involved very large populations to get any kind of statistical data that would confirm or validate that these products are actually active. So from our perspective, we believe that this marketplace is ripe for product differentiation. We've met a lot of our clients who have expressed a deep interest in new and more effective broad-spectrum products. And this is what we are presenting today in terms of our development program. Our objective is quite simple. We really want to develop and commercialize a new generation of immunostimulants. And you can categorize these into basically two categories. There are water-soluble and there are water-insoluble antigens. So just to give you an idea, a very good example of a water-insoluble particulate antigen is something based on yeast beta-glucan. That product is very insoluble in water, and yet it's a very effective product. So what we've done over the past year is we have scoured the world for opportunities to in-license antigens that are new, or immunostimulants that are new to the U.S. market, that might offer us the benefits that we're looking for and our clients are looking for when they in-license the products that we develop for them. From a product design point of view, all we're doing is trying to keep your overall health in mind. And of course, one way to do that is to optimize the innate and adaptive system response. And as when Dr. Gita Jensen gets up and talks, she's going to talk a little bit more about this, but there are many, many factors which deplete your immune system. Principal among them are exercise recovery, stress, travel exposure. Here I have a picture of the Las Vegas line that you're going to be in the next day or two, waiting to get through security. And of course, chronic fatigue is one of the main complaints that women have when they go to visit their general practitioners. And one element of chronic fatigue, not all elements of chronic fatigue, are sleep deprivation. And sleep deprivation is known to cause a dramatic reduction in your immune system. So basically, you're going to hear some very interesting results from our preliminary screening of the in-licensed products that we are looking at to develop and to also provide validated clinical trials on. Some of the things you may hear today include things like dendritic cells, natural T killer cells, T cells, T helper cells. You'll hear things like the word cytokine. You'll hear things like complement receptor. And for most of us, including myself, it has been a tremendous educational process for me to understand exactly how the immune systems work. So without further ado, I would like to introduce you Dr. Gita Jensen from NIST Labs, who is collaborating with us on this overall program. Gita.
Thank you. Can you all hear me? Well, it is really quite simple. The immune system is fast. When you hear about the innate immune system and the adaptive immune system, think fast, think innate, immediate, adaptive, long-term. When we recognize a pathogen, the innate immediate immune response will produce an immediate counterattack. Immediate, we're talking seconds, minutes, hours. While those cells are busy producing an immediate defense, other cells are chewing the invader up to bits and presenting it to other immune cells to provide long-term protection and cytokine production, immunoglobulin production, and long-term memory, such that next time we may meet the same invading pathogen, we will respond even faster and even more efficient. So how can a nutrition supplement, an oral supplement such as the Immunum program, affect the human immune system? This is a very important point in talking about consumable immune supportive, immune modulating supplements. This is key because it is not necessarily important whether all the compounds enter into the bloodstream. This is different than when we talk about vitamin C, we talk about zinc for the immune system. This is a different way of thinking, it's a different function. We are not talking so much about bioavailability, we are talking about bioefficacy. There are many sequential steps. There are the antigens and the immunostimulants interacting at the cellular level with cells already in the gut mucosa. There are downstream cascades of cytokinetics affecting the whole body, and there are even nerve signals coming from the gut to distant tissues. And again, it's happening fast. So let's have a quick look at the gut mucosa. The yellow cells out in this is the mucosal barrier. What is above is where the food and the probiotics and everything else hangs out. And below is the actual immune tissue in the gut mucosa. I've given two examples of different types of consumable immune products. Some little black squares up here and some orange squares over here. I want to use this to illustrate two potential different mechanisms. The little black squares are actually taken up through specialized cells, are chewed up into little pieces and presented to immune cells underneath, cells that will then interact with other cells and trigger cascades of cellular and cytokine-mediated responses. Now these little orange squares is an example of an immune supplement that may actually never enter our bloodstream. Yeah, John mentioned dendritic cells. They are really important cells in searching out foreign substances and triggering reaction. This example is one I really like. If you have a dendritic cell sitting inside the gut barrier and it's sticking tentacles out between the gut barrier cells right into the gut lumen and sampling antigens in the food we have eaten. These particles may never enter, but the dendritic cell is now altered. It has received a signal through its receptors, and the cell body in the mucosal immune tissue will interact with other cells and trigger cytokine cascades and nerve signals. So, with a fast immune response, how does that dictate our clinical trials, and how are the most optimal ways of proving that a consumable immune supplement is actually working? Again, we have to think fast. One of the study designs we like to use for immune supplements that actually work is what we call an acute trial, where we are looking at responses within a couple of hours. Since the people we involve in our studies, we can't throw viruses and bacteria at them and study their fever and study their responses. It's not what we want to do. But we can feed these people a single dose, and we can take blood samples at different time points. We can take that out into the lab, and we can look at changes in immune surveillance and the priming of the cells. So when we take the cells to the laboratory, and we then can trigger an insult by what looks like a virus and what looks like a bacterium, how are they responding? Are they primed to respond more effectively? So here's a person in a study having the blood drawn. The blood cells are being taken through flow cytometry, where we electronically can filter out and analyze the amounts and proportions of different cell types of the immune system. And at the same time, we are taking the blood plasma, the non-cellular fraction of the blood, and we are doing multiplex analysis of, for example, pro- and anti-inflammatory cytokines and reparative growth factors. These clinical trials determine immune surveillance, immune cell activation, changes to cytokine levels, and as I mentioned, immune cell priming is really an important factor. Can we that rapidly measure changes to responses to pathogens? Now let's take a step back and talk about the importance of screening of potential ingredients in immune formulations. We use human immune cells, we culture them, and we expose them to a very wide range of the potential ingredient. In what I'll show you in a few slides, we are actually looking at a million-fold dose range. We are looking at the immune cell activation, and we are looking at the cytokine production. So the cytokines, again, being the mediators of long-distance and throughout the body communication of what is going on. And we are looking at both activating pro-inflammatory and resolving anti-inflammatory mediators. So we need to be able to turn on and off the heat of the immune system. We need to go from a resting state to a very, very active defense mechanism very, very fast. And we need to eventually be able to return to normal alertness, but without so much aggressive responses. So down here in the bottom is a diagram of a normal immune response. We will see a very, very rapid activation, and we will see a resolution. If things go wrong, and we see that rapid activation, but we don't see the resolution, that is where we have chronic inflammation. And you've all heard about chronic inflammation, both in metabolic syndrome, in type 2 diabetes, cardiovascular disease, age-related cognitive decline, depression. So it's important to both activate and resolve, also when we are looking for ingredients. We'll take a little peek at some examples of ongoing research. Want to show some comparative dose responses of 8 potential ingredients. Here we are looking at natural killer cell activation, so the cells that are important in antiviral and anti-cancer defenses. And down here we are looking at T cells, more regulating types of cells that are also very important in the cytokine production. So you will see a very, very big difference between these ingredients in how they're triggering cellular responses. You will see quite a few interesting candidates here in terms of natural killer cell activation, and they are also playing out on the T cell activation end. Over here you will actually see that many of the potential ingredients are inhibiting a certain aspect of T cell responses, and even at very low doses. Again, this is a million-fold dose range. And then we're using advanced multiplex technology to look at simultaneous analysis of several dozens of cytokines, regulating, activating, and resolving cytokines. This is a really important piece of data. In this kind of screening, we are looking at 2 pro-inflammatory activating cytokines, and again across this very broad dose range. And we are getting some extremely clear yes and no answers at this level. We are seeing that 4 of the 8 potential ingredients have no measurable effects on these 2 important key cytokines. The other 4 products actually had quite robust activating effects with very large amplifications of these cytokines. The top one is the interferon gamma, very important in also activating natural killer cells, and MCP1, another monocyte macrophage involved factor up here. Very important dose response. If we had just done a human clinical trial on these doses, the products could have come up very similar. Now, the importance of this is that more is not always better. And the maximum dose that you think you can afford to put into a blend or put into a formulation may not be the optimal dose. You will see in this case that you can actually go 1,000-fold down and in dose in this particular screening assay and see a very, very robust response in some of these ingredients. This allows you to maybe make room for more products in your blend and still reach an affordable product that works. Now, interesting, as I told you, we both need activation and we need resolution. We are looking at two cytokines as an example for factors that help resolve that reaction, that activation, and return the immune system back to normal. And again, we see that we get some clear yes-no answers. We are seeing that the same 4 ingredients that help activate also help resolve. And this is the thing to look to when you're planning a blend. You will also see that that really leaves 50% of these potential ingredients with no measurable effect in this particular assay. So the strategic process of discovery and validation is quite straightforward. We got to identify the candidates. We got to also evaluate their activity after we have mimicked the digestive procedure and make sure that when they've been ingested and digested, there's still activity, maybe even more activity. And then we need to test the acute rapid effects in humans and validate doses, and that we also need to validate that when we start blending, we are not just seeing as a competition of how ingredients are working, we are actually seeing synergy. Once this whole process has been completed, that validating process, then we're ready to move into the more claim-specific, randomized placebo-controlled human trials, for example, against cold or flu, against allergies and other immune problems. So, yes, it's complex. The path is simple to get you there fast.
Thank you, Gita. And I had mentioned before, Gita is the owner and also the R&D director at NIST Labs. And of course, she's a PhD immunologist. Hopefully, we've been able to simplify immunology for you and also help you understand what stage of clinical development we are in at this point in time. So we've in-licensed quite a few potential antigens. We've gone through the initial in vitro screening using human cells, and we are now ready to move on to the next steps in the overall program. So always we're thinking about how we can now license and market these products in the real world. And every time we do clinical trials, we think about the marketing aspects as well as the regulatory aspects and, of course, the activity of these various compounds. So we look at this from three different perspectives. First of all, we look at it from an efficacy point of view. We must have a meaningful measure of the efficacy of these products at the cellular immune proliferation level, at the immune surveillance level, and also at the whether or not these products can cross-talk with other cellular components in the immune system. And we need to have a meaningful comparison between the standard of care that is available in the marketplace today. And this can all occur very, very rapidly. And finally, we want to use modern technology to ensure that we're doing the best available, we're using the best available tools to ensure that we are accurately developing these products. And you heard things like flow cytometry, cytokine profiling, and ex vivo challenges. There's no longer a need to run a one-year trial on a large population of people to determine whether or not you have a commercially viable product. So, from an overall perspective, we are bringing our in-licensed clients unique immunostimulant offerings. And we're doing this through a focused stage gate product development program where we develop products and or formulations that have protected intellectual property and or protected access to the various products. And we can also combine these various products to ingredients to provide, as Gita said, complementary broad-spectrum performance. And this is how we can leapfrog the existing standard of care products in the marketplace. And obviously there are many available presentations, formulation presentations that work, and in fact some of these cytokines are very specific for the type of formulation presentation we would like to develop from a clinically validated point of view. So we'll be able to offer quite a variety of clinically validated formulations. And most importantly, we're going to be doing this in a regulatory environment that we understand very well and with its limitations. So thank you very much, and Guy, thank you very much for joining us for this presentation. If you have any questions, be happy to field them or have Guy to field them for you. All right, thank you very much for your time.